Good morning. I want to continue in this video on chapter 12. Specifically, I want to talk about the contributions of Gregor Mendel and some of Mendel's rules and concepts that he developed from working with the garden pea plant. Mendel was an Austrian monk. He worked for at least a decade or so on the inheritance patterns in the garden pea. He published his work in, I believe, 1866. He became an administrator in the church after that and passed away, I believe, in 1884. His paper that he wrote and published wasn't discovered, I believe, until 1901 by Thomas Morgan and some other American geneticists, and they really, tried to repeat some of Mendel's findings, and really kind of then gave him credit as being the father of genetics, the father of inheritance from his work with the garden pea. What you see in the photograph is how pea plants produce seeds, pollen from one plant can be deposited onto the stigma of another plant. In this case, you have pollen from a purple flowered plant being deposited onto the stigma of a white flowered pea plant. That white flowered pea plant would have its eggs fertilized by the sperm in the pollen. It would form seeds within its ovary, and then when planted, those seeds would grow up, and lo and behold, the dominant flower color of purple would be shown in the first generation offspring. So the purple and white flowers are the parents, and the offspring are the F1. Mendel's experiments provided some evidence for the inheritance of characteristics and their genetic basis. At or before that time, the inheritance of characteristics was thought to be due to a blending between the parents. So if you have two parents, their offspring should show a intermediate set of characteristics. That's what I mean by blending. Mendel showed that traits were governed by specific units of inheritance that he called factors, and we know that these factors are now referred to as genes, as the units of inheritance. Mendel came up with some very specific ratios for the mating experiments he did with the garden pea, and he came up with some laws of inheritance that really laid the foundation for what we call genetics today. Mendel performed a whole set of series of crosses with the garden pea, Piosum sativum. One set of crosses he did was referred to as monohybrid, where he examined one trait and followed it through several generations. He looked at two traits, referred to as dihybrid crosses, and followed those through several generations. And he did test crosses, where you have one parent that you do not know its genotype, and you cross that parent with a parent that is homozygous recessive. By examining the offspring, you can then determine the genotype of the unknown parent. Mendel examined 7 different traits in the garden pea plant. Conditions of the seeds, of the fruit pods, of the flowers, of the height of the plant, and of the position of the flowers. So easily observable characteristics. We refer to these as phenotypic characteristics or phenotypic traits. A fellow by the name of Reginald Punnett, he designed the Punnett square, if you will, as a methodology for solving genetics problems. And one of the most difficult things that students have to do with figuring out the answer to a genetics problem is how many gametes does a parent produce? So if a parent has two large A alleles, it can only produce a gamete that contains a large A allele in that gamete. A parent with two small A alleles can only produce gametes with small A alleles in those gametes. So that means if a parent is homozygous for a trait, there's only one gamete combination possible. If a parent is heterozygous, there are two gamete combinations possible. If a parent has a big A and a little a, then it's possible for gametes to contain the dominant A, capital letter A, in its allele, or the lowercase recessive little a in its alleles. So in example 1, big A, big B, there are two possible gamete combinations, big A, big B, and little a, big B. So if you have two different alleles, in this case A and B, then there are two different alleles in each gamete, A and a B. In number 2, big A, big B, little b, there are 4 combinations possible of A and B. And in number 3, you have a third allele, letter C, added. And there are 8 combinations. 2 times 2 times 2 is 8 different combinations of A, B, and C. So you would have 3 alleles possible in a gamete for number 3. So determining gamete combinations is real important for solving genetics problems. In the example below, the probability for a round P shape is 75%. The probability of a wrinkled P shape is 25%. So probabilities are used to determine the likelihood of some occurrence. The Punnett square is used to determine the outcomes of a mating. So if 2 round seeded pea plants are mated, those are your parents, and if their genotype is heterozygous, the likelihood of their offspring having round seeds is 75%, and the likelihood of their offspring having wrinkled seeds is 25%. So that is the use of the Punnett square for working a genetics problem, and the outcomes are often given as probabilities. How likely is this outcome to be? When Gregor Mendel performed monohybrid crosses, he started out with parents that were homozygous. His first generation offspring were all heterozygous and illustrated, or they all had the dominant trait. 100% likely was the probability. So he would take 2 first generation offspring and he would interbreed them. And when he did, his F2 offspring had a probability of 75% dominant trait and 25% recessive, or a 3-to-1 ratio. Here you see flower color with 100% of the first generation having purple and 75% of the second generation having purple and 25% of the second generation having white flowers. This particular data set shows all of Mendel's monohybrid crosses and shows the approximate 3-to-1 ratio in the second generation, whether it be for seed shape or stem height or length. You might notice that he grew several thousand plants, and it took years to grow these plants and calculate the different ratios of these phenotypic traits. So this was a very long-term experiment with a very large data set. From monohybrid crosses, Mendel came up with several laws. One was the law of dominance. The law of dominance is that there are alleles in a population that their gene product, their protein product is always produced, and those particular alleles are referred to as dominant alleles. So the law of dominance is one of Mendel's rules. The law of segregation is another, and it states that if you have two alleles for a trait, those alleles will separate or segregate and end up into separate gametes. So if you look at the bottom combination of capital letter P and lowercase p, those two alleles, since they're different, they will separate from one another and they will not end up in the same gamete at the end of meiosis. So you have gametes with capital letter P and gametes with lowercase p only. You do not have a gamete with both a uppercase letter P and a lowercase letter p. So the law of segregation is one of Mendel's rules, as is the law of dominance. Mendel performed test crosses to determine genotype of an unknown dominant parent, and it provided him with more support for the monohybrid predictions. So if you have an unknown purple flowered pea plant, you mate it with a recessive. If the unknown dominant individual is homozygous, all of the F1 offspring will show the dominant trait, as you see in the box at the bottom. If the dominant individual is heterozygous, half of the individuals will show the dominant trait and half will show the recessive trait. So test crosses were very useful in determining dominant parent genotypes and for providing support for the monohybrid predictions of 100% or even the 3-to-1 ratios that he came up with. With a dihybrid cross, Mendel looked at two traits and how two characteristics could be inherited. Again, if he started with homozygous parents, in this case you have round and yellow crossed with wrinkled and green seeds, the first generation would, with 100% probability, show the dominant traits, round and yellow. When he would then cross two of the first generation and then see the results of growing those seeds, he found a 9-to-3 to 3-to-1 ratio in which 9 of the 16 offspring possibilities were dominant for both traits. 3 were recessive for one and dominant for another. 3 were dominant for one and recessive for the other. And 1 of the 16 was recessive for both traits, round and wrinkled. And you can see that in the Punnett square that shows 16 different boxes. So you've got a 9-to-3 to 3-to-1 ratio. And the experimental data in the top right of 315 going to 32, those were his numbers, and they approximate that 9-to-3 to 3-to-1 ratio pretty well. The same would be expected if you took two other characteristics such as tall and purple flowers and dwarf and white, just as a second example. Mendel's theory of independent assortment came from dihybrid data and states that if you have two alleles, A's and B's, they will sort into gametes independently of one another. So you have a possibility for big A, big B, big A, little b, big B, and little a, little b. You've got four different